Hello, thank you for joining me for the 3MT 3-Minute Thesis 2020 final competition at the University of Florida. We appreciate you taking the time to watch the presentations today. To give you an overview of the competition, 3MT began as a research communication competition developed by the University of Queensland in Australia. It challenges PhD students to compress their research into 3 minutes and present a compelling oration to a non-specialist audience. It began in 2008 and has since become an international competition. The UF 3MT competition is held each year in September and October. Our rules are slightly different this year as we've had to transition to a virtual competition. They are as follows. Each student is allowed a single static PowerPoint slide, and they may use no additional props. The recordings they have submitted are limited to 3 minutes and 10 seconds, and they are to be spoken word only. The decision of our adjudicating panel is final. Next, I'd like to introduce you to our judging panel. This year, we have Dr. Samisha Barnes, the Director for the McNair Scholars Program, Dr. Kristen Galvin, the Assistant Director for Graduate Engagement in the Center for Humanities in the Public Sphere, Melina Jimenez, teacher with the English Language Institute, Dr. Shobha Jaishankar, Assistant Vice President and Lecturer in the Office of Research, Dr. Betty Loisel, Director of the Tropical Conservation and Development Program in the Center for Latin American Studies, and Dr. Michael Kung, Director of Global Education and Program Director for the Sustainable Design Master's Program in the College of Design, Construction, and Planning. Our judges will review the presentations based on 2 sets of criteria. The first is comprehension and content. Did the presentation provide an understanding of the background and significance to the research question being addressed, while explaining terminology and avoiding jargon? Did the presentation clearly describe the impact and/or results of the research, including conclusions and outcomes? Did the presentation follow a clear and logical sequence? Was the thesis topic, research significance, results and/or impact and outcomes communicated in language appropriate to a non-specialist audience? Did the presenters spend adequate time on each element of their presentation? Or did they elaborate for too long on one aspect, or was the presentation rushed? Our second set of judging criteria is engagement and communication. Did the oration make the audience want to know more, and was the presenter careful not to trivialize or generalize their research? Did the presenter convey enthusiasm for their research? And did they capture and maintain their audience's attention? Did the speaker have sufficient stage presence, eye contact, and vocal range? And finally, did the PowerPoint slide enhance their presentation? Was it clear, legible, and concise? Now that we've introduced our judges, the judging criteria, and the rules, I'd like to present our 2020 final presentations.
Close your eyes for a moment and visualize the effects of climate change. When I do this, my mind immediately jumps to polar bears stranded on dwindling ice sheets or forests engulfed in flames. But today, I'm here to talk about an effect of climate change that we can't usually picture in our minds, an effect that is much more insidious. I'm here to discuss how climate change alters the timing of biological events. Previous research links climate change to shifts in the timing of biological events such as bird migration and when plants begin to produce new leaves. Climate change causes such shifts because plants and animals rely on abiotic cues such as temperature and rainfall to tell them when they should do certain activities during the year. When these cues change, for example, when temperature rises, so changes the timing of certain biological events. My research investigates a particular example of this. Specifically, I study how climate change affects the relationship between plants and bees by altering the timing of plant bloom and bee activity. This is particularly important because the relationship between bees and flowering plants is critical for human food production, because bees do the essential role of pollinating flowering plants, thereby allowing them to sexually reproduce and grow the plant tissues such as fruits that we love to eat. Common commodities like almonds, blueberries, and watermelons all depend on bees for pollination, and without them, production would decline precipitously. But in order for bees to pollinate plants, the timing of bee activity must overlap with when plants are producing flowers. However, for many bee and plant species, these events occur for a very limited window of time during the year, and plants and bees rely on the precise overlap of these events in order to continue to grow and reproduce successfully. The aim of my research is to determine if the timing of plant bloom and bee activity are shifting in response to climate change, and to evaluate how quickly these shifts in timing are occurring. To assess this, I go back in time by gathering information from long dead plant and bee specimens housed in natural history collections to determine what the timing of bee activity and plant bloom was in the past and compare it to what it is today. My research finds that, indeed, the timing of plant bloom and bee activity are both shifting in response to climate change. However, the timing of plant bloom is shifting more quickly than the timing of bee activity, which may result in even less overlap between these biological events in the future. This finding indicates that the effects of climate change can negatively impact not only charismatic polar bears and forests in far-off places, but also relationships between organisms like plants and pollinators that are integral to human well-being.
Hello, I have bad news. In the United States, every year, over 1 million people are hospitalized with a diagnosis of heart failure. For each patient, every day during their hospitalization, various types of data will be generated, like labs, medications that they take, hospitalization characteristics, medical images, vitals, demographics like age, gender, and social behavior. Now imagine you are a physician and it's time to make a decision whether the patient is ready to be discharged or the patient still needs medical care at the hospital. How would you make such a decision? Let me tell you something. It's almost impossible to do so, considering all the changes the patient might have during their hospitalization. If you discharge a patient who is still at a vulnerable stage, he might come back with a more severe condition, or he might die before readmission. On the other hand, if you keep a patient who is doing well at the hospital, there's a substantial cost for the patient and the hospital. My study aims to help physicians to make such decisions. In our study, we collected data for 3,189 patients. These patients were admitted to University of Florida Shands from 2011 to 2019 with a diagnosis of heart failure. Among them, 15% were readmitted due to acute heart failure, and 11% unfortunately died within 90 days after discharge. We processed this data using data analysis tools. Then this data were fed into a machine learning model. We utilized a logistic regression model. Why this specific model? Because this specific model can provide us very helpful information to identify significant risk factors associated with readmission status. This model can help physicians in assessing patients' risk and classify them as high or low risk. Using this model, we can decrease the rate of readmission by providing on-time interventions or follow-up visits to high-risk patients. On the other hand, we can decrease cost by providing routine heart failure care to low-risk patients. This model appears to be more discriminative compared to commonly accepted models in the literature. And also, this risk assessment model is broadly applicable to care of heart failure patients. Thank you.
Just for a few seconds, imagine if there was no COVID-19. It's fall, you're a season ticket holder, and you're very excited to walk into that crowded stadium with your friends for the first Gator game. Feels good, doesn't it? Now think about Bob. Bob didn't have to wait for a pandemic to feel restricted and dissatisfied with his outdoor walking abilities. Bob, like 800,000 other Americans, suffered from a stroke last year, rendering one side of his body weak, limiting his walking abilities. Although Bob is able to walk safely in his home environment, he really values walking outdoors so that he can resume his weekly shopping trips with his granddaughter to Oaksmall. Constantly staying at home has also made Bob very sedentary. You probably must have heard that you need to take 10,000 steps in a day to maintain a healthy lifestyle. And you probably use a smartwatch or a Fitbit to find out how many steps you take in a day. Well, Bob only takes about 4,000 steps in a day, which is just half of what other healthy older adults of his age take in their daily life. This puts Bob at a risk for a second stroke. Bob's therapist really wants to encourage him to walk outdoors so that he can be more active and independent in his daily life. But wait, there is one major problem. Where did Bob take these 4,000 steps? Just with the activity monitor alone, we don't know the location of his steps. Was he walking in circles in his kitchen or garage? Was he walking in his backyard? Or was he walking at Oaksmall? My dissertation is going to help solve this problem. We use a GPS device almost every day while driving to new locations. We are going to combine this activity monitor with a GPS device to see how far away from home are individuals with stroke walking in their daily life. By combining these two devices, we will be able to differentiate between home steps and community steps for each individual participant in our study. Let's see what we have found out till now. In this graph over here, I have 9 healthy older adults depicted in red and 22 individuals with stroke depicted in green. As compared to healthy older adults, individuals with stroke took 32% fewer steps in their home. But more importantly and more concerning is that they took 74% fewer steps in their community. This signifies the need to specifically measure and ultimately improve community ambulation for people with stroke, as it has been a long and challenging process for people like Bob. Thank you.
Long live the king. King of the herbs, that is. The plant name basil comes from the ancient Greek basilius, which means king, and what a fitting name for the most popular herb in the world. This plant that we use to top our pizza with is packed with flavor and health-promoting compounds. Basil bred by the University of Florida has another important trait: natural resistance to downy mildew, a plant disease that continues to devastate basil and other crops. Actually, up to 40% of the food in our fields are destroyed every year by pests and pathogens just like downy mildew. UF basil has high amounts of health-promoting compounds, great flavor, and stands up to downy mildew without the need for any chemicals. But how does it do it all? The answer is inside its DNA. Quick genetics lesson. DNA is like the cookbook that every cell must read in order to function. Inside these cookbooks are genes or recipes that are used for different traits or meals. My thesis focuses on comparing the cookbooks of UF basil to other basil plants cookbooks that are not making the same fantastic meals. A basil's cookbook is massive, so these comparisons produce pretty long lists of different recipes that might be used for these different meals. In order to test if one of these recipes is used in a certain meal, we can use gene editing to cut out that recipe from the plant's cookbook. If the gene-edited plant does not make that same meal, then we know that the gene that we cut out is responsible. Once we identify the different genes used by UF basil to be downy mildew resistant, produce high amounts of flavor and health-promoting compounds, We can transfer these recipes into the cookbooks of other crops so that they also can make these fantastic meals. Imagine healthier corn, tastier broccoli, and getting more out of our fields every year. Crops that do better in the field and are better on the plate can have significant benefits to both people and the planet. When we take what we know from the king of the herbs and apply it to other crops, it can go a long way in meeting the challenge of producing more with less and providing outstanding fruits and vegetables to everyone around the world. Thank you.
Hello, my name is David Campbell, and I'm a PhD student at the University of Florida in the Horticultural Sciences Department. I'm pleased to present research that was built on the shoulders of giants that's intended to help Florida farmers and consumers. Peaches originated from China. Peach trees grow in the north and the south, but only trees in the colder north produce fruit that resembles what we eat today. After trade and transport and colonization, peach was established in Georgia and South Carolina. The left part of this slide shows the transition that was started in the 1950s. Breeding efforts by top UF scientists, Drs. Ralph Sharp, Wayne Sherman, and most recently Jose Chaparro, allow us to do what we could have never done before, grow peaches in low chill environments such as Central Florida. At the same time, organic production was increasing. Year after year, the number of farms, acres, and value of organic produce has been increasing. In 2017, I was pleased to join a team of top-notch researchers at Clemson, Georgia, and Florida to help farmers in the Southeast grow organic peaches. Organic peach farmers in the Southeast will regularly lose over 50% of their crop to arthropod pests, such as plum curculio and stink bugs, as well as fungal pathogens that cause brown rot and scab. In order to help these farmers, we conducted an experiment to protect fruit by enclosing them in a bag to provide physical protection. And after 2 years of evaluation, we found that the bags worked. Peaches grown in bags had a delayed maturity, but most importantly, were less likely to have brown rot at harvest and post-harvest. They had less scab, and in 2018 protected the fruit from an opportunistic Drosophilid fruit fly. Given these positive results, we decided to take the next crop of peaches to a concert. Well, not really, but we did continue the research by relying on photoselective inserts that are identical to the ones used in concerts and movies that adjust the light quality. The figures on the bottom right shows a peach grown in a green bag with sensors inside the bag and spectrums for each level that we studied: blue light, green, red, and black. The peaks of each treatment correspond to the activity or inactivity of at least one photoreceptor. Now, photoreceptors are like antennas tuned into a particular wavelength, and when active, produce biochemical changes in the fruit, such as increased anthocyanin content. For example, phytochrome and cryptochrome, with peaks in the red and the blue range, have been shown to increase anthocyanin content. And in one experiment, peaches grown in black bags also increased the anthocyanin content. Increases in anthocyanin, which is an antioxidant, will make food more nutritious and potentially more marketable. We conducted these experiments to build on the past legacy that brought peach production into Florida, to support organic farmers and consumers, and find ways to produce abundant supplies of nutritious food. Thank you for your time.
What comes to your mind when you hear the word opioid? I bet you will say the opioid epidemic, which is killing more people than car crash every day. At the same time, opioid is an extremely effective painkiller that might be the last resort for millions of patients who are suffering from pain. Imagine yourself as a doctor. Will you prescribe opioids to patients whose pain only responds to this medication? What if I tell you there's this drug called gabapentin that has shown so many promises in relieving pain while reducing opioid consumption? Would you like to add this gabapentin to the traditional opioid therapy? If your answer is yes, then you are among thousands of clinicians who are using this as opioid alternatives. And because of that, gabapentin prescriptions have skyrocketed in recent years. In fact, it has become the 6th most prescribed medications in the United States today, with 68 million scripts written annually. So, if gabapentin is truly as good as people believe, the opioid epidemic might have a solution. right? However, more and more case reports accuse these drugs of causing breathing difficulties, drug overdose, and traumatic injury. Also, there's no answer as to whether they indeed reduce opioid consumption in patients undergoing major surgeries. So, in my dissertation, I, just like Sherlock Holmes, trying to solve this case by evaluating the safety and effectiveness of gabapentin. I conducted 3 observational studies using health records from 400,000 opioid users. I divided patients into 2 groups. One group used both gabapentin and opioids, and the other group used only opioids. Then I compared the incidence of adverse events and opioid consumption between these 2 groups, holding other factors the same. Contrary to many people have believed, I found gabapentin guilty of increasing the risk of opioid overdose by nearly 40% and traumatic injury by more than 50%. Also, I did not find them to reduce opioid consumption to a clinically meaningful level. This means gabapentin may not be effective in reducing opioid consumption in the surgical population. Instead, they could increase the risk of severe adverse events. Therefore, the use of gabapentin as opioid alternatives needs extra caution. After all, we do not want to put patients at high risk for unintended consequences, right? By doing my dissertation, I provided clinicians with real-world evidence on the benefit and the risk associated with gabapentin. So let me ask that question again. Will you prescribe gabapentin and opioid together to those who need opioids? Thank you for your attention.
Have you ever wondered what happens when you fall off your bike and hit your head? Or when a football player kicks another one across the head thinking it was the ball? What happens to the energy from that impact? Well, if you're wearing a helmet, that energy will go through the material that's inside your helmet, keeping your brain safe and impact-free. This, however, is not usually the case, as the current material, polyethylene covinyl acetate, or EVA for short, does not do that great a job. This can be seen in the plot of impact energy versus temperature. This plot will tell us how well a material will absorb energy at a specific temperature. Now, EVA does a great job at absorbing energy at around -10 C. But at intraoral temperatures, 37 C, which is the temperature inside your body, you actually see a dip in its ability. This property is unfavorable, and it's why we still have concussions and brain injuries and long-term brain damage normally seen in professional athletes. This is where my work comes in. Damping materials are everywhere. We already discussed helmets and mouth guards, which are normally used in hotter temperatures. But what about lower temperature activities, like a sound room? This calls for a material that can work great at many temperatures or be modified depending on the application. Thiolenes are composed of a thiol molecule and a vinyl molecule that, when put together, form flexible thiol ether bonds between the 2 ends. These have already been shown to do a better job at dissipating energy through molecular motions than EVA. Now, what we want to do is incorporate a third component, a spiropyran molecule, that will not only do a better job, but also impart additional function. This picture here is to help you imagine it on a microscopic level. These blue lines are the thiolene, and the orange and yellow are the spiropyran. Upon impact, the strained spiropyran molecule will break, allowing the remainder of the network to maintain its integrity. A second very cool function is that upon cleavage, these molecules are colorimetric, so you will actually see a color change from point A to point B. And the third is that this fracture is actually self-healable through UV light. Now, the best way to summarize all this is with the following story. Let's say you send your child out to ride his bike, and while you're not watching, he falls off his bike and hits his head. Well, you have nothing to worry about because all of that energy will be absorbed by his helmet. Also, when he comes inside, you will be able to see if and where he hit his head because the impact points will actually be in purple from the colorimetric spiropyran. Finally, you will be able to fix the helmet yourself, giving you a recyclable helmet that does not compromise your child's safety.
Before I begin, please do not be intimidated by all the molecules. I know that chemistry can be scary. Organic chemists take the main inspiration for what we do from nature. Nature is incredible in the way that she takes the simplest chemical fragment, such as the isoprene unit shown at the top of the slide, and uses enzymes to build highly complex molecules. Enzymes are necessary to transform this unit into the target molecule and also add other functional atoms, such as nitrogen and oxygen. More importantly, nature creates molecules that have been found to be paramount to the improvement of human life. The molecules on the slide are some of the more famous examples of that, such as paclitaxel. Paclitaxel is quite notorious among organic chemists. It is isolated from the bark of the Pacific yew tree, and it has really strong anti-cancer activity and is currently being used in chemotherapy treatments. But it has been historically difficult for chemists to make it due to the complexity of its structure. Since we cannot efficiently and humanely extract enough of these types of compounds from living organisms such as the tree in quantities that can be useful to us, chemists come in with the task of making them from materials that we can access and buy. We want to create methods that accomplish what enzymes can do so elegantly, but with our limited resources. So as an organic methodology chemist, I am interested in new chemical transformations to make analogs of these natural products. Analogs are basically molecules that are similar to the main molecule we're interested in, usually bearing the same main green core that you see highlighted. Analog synthesis is also essential because analogs have been shown to demonstrate similar and sometimes even stronger biological activity as the exact molecule target. The main challenge for us is synthesizing analogs in new and efficient ways from very cheap and abundant starting materials. I started my project with cheap chemical building blocks costing about $150 per kilogram and developed an efficient route to the intermediate you see in the middle. This intermediate is valuable because depending on the chemical conditions that I continue to subject it to, I can manipulate it to make two different molecules. We can make percyanyl analogs or ramipril analogs. Ramipril is another currently marketed drug to treat a variety of cardiovascular diseases. With my newly discovered and developed chemical route, we can synthesize a variety of these analogs. The hope for my chemistry now is not only that I have contributed new chemical methods to my field, but also that these analogs can potentially have similar biological activity to its parent natural products. I hope I have given you a glimpse into the elusive world of the chemistry laboratory and that you have gained some appreciation for how challenging the work that we can do can be. And thank you.
Many of us know of someone that is fighting cancer. Some of us may have lost our loved ones to cancer. While screening, early detection, and advancements to treatment have improved cancer survival, cancer still remains to be one of the leading causes of death in the United States. But why is that the case? Let's look at breast cancer as an example. Breast cancer patients generally do not die from primary cancer confined to the breast. Almost 99% of patients survive with this localized disease. However, patient survival decreases with regional disease and significantly with this distant disease. This process of cancer dissemination from primary organ to secondary organs is called cancer metastasis. Breast cancer can metastasize to the brain, liver, the lungs, and the bone, with bone being the most common site of breast cancer metastasis and is the focus of my dissertation project. Not surprisingly, bone metastases severely impact the quality of life for these patients through inflicting bone pain, fractures, and nerve compression. And unfortunately, these patients with bone metastases are generally no longer considered for curative treatment and only receives palliative treatment. My dissertation project focuses on identifying new treatments that can impair cancer dissemination and also prevent active bone remodeling. For this, I turn to understanding the cellular communications and interactions between breast cancer cells and cells in the bone. These cells communicate through signaling pathways. In normal bones, there is a healthy balance and regulated signaling between cells that chew up the bone, known as osteoclasts, as shown in red, and cells that build the bone, known as osteoblasts, shown in blue. However, in cancer cells, many signaling pathways are hijacked to promote cancer development. When breast cancer cells reach the bone, they disrupt this healthy balance and signal the osteoclasts to further break down the bone so that tumor cells can grow into these bone cavities, as shown by the white arrows. This breakdown of bone is also the main reason for skeletal-related events and lower quality of life. I have identified a therapeutically targetable signaling protein that is present in both cancer cells and osteoclasts, and that facilitates metastatic behaviors and bone remodeling. I hope that my dissertation project can help develop curative therapies and improve quality of life for patients with bone metastases. Thank you.
Hi, did you watch the Chernobyl series on HBO? If you did, then you might remember that in the first episode, there is an explosion followed by discussion on hydrogen. So where is the connection between hydrogen gas and nuclear power plant? Well, if you look at the heart of a nuclear reactor, we have fuel pin. Fuel pin contains the fuel rod, cladding material, and coolant water. Cladding material is responsible for transferring heat from fuel rod to coolant water. At Chernobyl power plant, zirconium alloy was used as a cladding material, like most of the power plants today, which is fine, but only that it's not. Because if the supply of coolant is shut off due to an accident, then the temperature in the cladding material increases drastically. And at high temperatures, zirconium reacts with steam to produce hydrogen gas, which could lead to an explosion, as shown in the Chernobyl series. In fact, this reaction has occurred at other places too. So, to address this problem, silicon carbide composite tubes have been identified as the candidate material to replace zirconium alloy. Silicon carbide composite tubes have potential to outperform zirconium alloy as a cladding material, and moreover, they do not undergo any dangerous chemical reaction at high temperatures. My research involves developing the failure envelope for silicon carbide composite tubes. Failure envelope gives a safe region within which the material will not break or fail. And at any point outside the safe region, the material is expected to fail. The challenge here is the manufacturing process is such that these composite tubes will not be same each time they are produced. It is understood that there will be some aspects of the material which will keep changing. So considering the uncertainty in the material parameters, what is the uncertainty associated with the failure envelopes is an important question. I intend to address that question with the help of numerical simulation. I will calibrate my model so that its result is same as that of the experiment for simple test scenarios, like a hoop test, wherein the pressure is the only force acting on the inside surface of the tube, or the tensile test, wherein the specimen is just stretched along its axis. I will then use my verified numerical model to predict failure for more complicated combination of loads. This will be helpful in understanding the response of material towards load. With sufficient understanding of material behavior, we can replace the zirconium alloy with silicon carbide composite tubes and eliminate the risk of hydrogen explosion. This will make the nuclear reactors more efficient and more safe than they currently are. And that's why the US Department of Energy funds my research. Thank you.
The UF Graduate School would like to thank you for watching our 3-Minute Thesis 2020 final presentations. We hope to see you in 2021.